So our last speaker in this kind of morning session is going to be Chuck Murry, who's joining us from UW Medicine, so just pretty nearby.
Good morning, everybody. It's a, wow, good-looking crowd today. Thanks to the organizing committee for inviting me, giving me a chance to speak here. And it's been really neat having this AICS as a neighbor across the street. And the science that's getting done here is compelling. And one of the unexpected benefits that we've seen, for example, is you are making our jobs in recruiting much easier, because there's a lot of faculty who are interested in coming to the University of Washington when they hear that there are potential interactions with the Allen Institute. So thanks for that also. What I'd like to talk about today is cells really as building blocks for tissue repair. I think a lot of us had our imaginations captivated, especially the elderly like myself amongst the crowd, when the stem cell revolution hit in the early part of the 2000s after the advances in Jamie Thompson's lab with human embryonic stem cells and so forth. So what I'd like to tell you about today is the work that we've been doing across the street to regenerate the heart using cardiomyocytes derived from stem cells. So the first thing I'd like to say is that heart disease is bad. You don't want it. It's really common and it's very expensive. That's basically what it amounts to. Is that it's the #1 cause of death worldwide now. And in the United States, it's the #1 cause that people get admitted in the hospital for. It's the #1 thing Medicare is expending money on. Probably direct care costs of about $250 billion a year in the U.S., and indirect costs exceed that, of course. And so it's a big one. Here's the rub. You start with a diseased coronary artery, and this is an atherosclerotic plaque. Typically, you get a blood clot or a thrombus that forms in it, and a chunk of the wall of the heart is not well perfused. Now, the heart has one of the highest metabolic rates in the body, very little in the way of energy reserves, and so as a consequence, cells start to die between 15 and 20 minutes after coronary occlusion. So the heart is the second most ischemia-sensitive organ in the body, second after the human brain, where you get about 5 minutes before cells start to die. So problem #1, the heart's very sensitive. Problem #2, the heart is about the least regenerative organ in the body. So if we look at a cross-section of the heart here, the left ventricle is the circle and the right ventricle is the crescent. A chunk of the wall is knocked off, and it heals by scar formation. So here's the problem. There's basically no cardiac regeneration. The heart heals by scar formation. And this is why a large number of people who develop myocardial infarctions go on to develop heart failure. Heart failure, as the baby boomers are aging out, is growing in epidemic numbers. And once you're diagnosed with heart failure, you have on average about 5 years to live. 50% population survival is 5 years after that. So it's worse than many kinds of cancer. Take breast cancer as an example, where on average people live about 11 years after diagnosis. So our notion is, can we somehow harness the power of stem cells to grow back the wall of the heart after a patient suffers a myocardial infarction? So this is kind of a sea shift. Medicine it rests on a couple of pillars. The first is really built on organic chemistry. I think this is why we torture pre-med students by using organic chemistry as the weed out course, right? Because it was small molecule chemistry that was really the foundation of pharmaceutical sciences. The second pillar came along when we started using cells, but we used cells as factories to build proteins, kind of like what David Baker was talking about, and using the proteins themselves as therapeutics, so antibodies, receptor antagonists, things like that. And then the third pillar that we posit will be emerging as regenerative science comes along is that cells themselves will actually be the therapeutic units. So let me give you a quick study on cardiac repair using stem cells. This condenses between 15 and 20 years into just a few bullet points. People have tried using adult stem cells to repair the heart for a long time, from bone marrow, from fat, from the heart itself. And the bottom line is these cells aren't very robust. They die within a week or so after transplantation. So they don't contribute as direct bricks and mortar type cells like we imagined at the onset. In contrast, if you take pluripotent stem cells and make them into cardiomyocytes and then transplant them, one can get long-term stable grafts of what we call new myocardium, new heart muscle, in the hearts of experimental animals. And we've worked our way up the food chain, starting in small animals like mice and rats, and then up into guinea pigs. And we found that this remuscularization of the wall enhanced cardiac function. One of the problems is this engraftment is a highly inefficient business. We're really, the vast majority of the cells die afterwards, and so getting complete remuscularization of the wall, where you really need mechanical oomph in order to restore the squeeze, that's been a challenge. And then the functional impact of putting human heart muscle into a large animal that resembles human cardiac physiology, that's really not been done anywhere. And so what I'd like to tell you today is 2 quick vignettes that talk about ways that we can enhance remuscularization per se, and then what are the effects of doing this in a large animal. All of this work is unpublished. That's not entirely by choice, but that's where things stand at the moment. So the first vignette I'd like to give you is work that's done by Caitlin Gerben when she was a graduate student in bioengineering in my laboratory. Caitlin is now gainfully employed by the Allen Institute for Cell Sciences. So that was also another unexpected benefit of you all moving across the street. Caitlin's project focused on using the notch system to control proliferation of cardiomyocytes after we transplanted them. The evil plan basically is if you can take a known mitogenic system for cardiomyocytes and deliver it to them in an intelligent, and deliver the cells with this signaling system as a mixed package, we should be able to use the compound interest of cell division to get better remuscularization. And so the way this works, people are probably familiar with the notch system. It's a ligand receptor system that involves a ligand presenting cell that has a transmembrane protein. Delta 1 is shown as just a prototypical ligand, and then the other cell is the receiving cell that has the notch receptor itself. There's receptor-ligand interaction, and this puts tension on the receptor, exposes a cryptic cleavage site that can be chewed off by a protease on the cell surface, and then this initiates a second proteolytic event on the inside of the plasma membrane, and it releases what's called the notch intracellular domain. This translocates to the nucleus and acts as a transcriptional regulator and activates transcription of a number of notch target genes, among which this has been well known to control proliferation of early stage cardiomyocytes. So the question is, can we harness this somehow to promote better heart regeneration? So first, in earlier work, we had developed a system where we could do this in 2-dimensional cell culture. So we took the delta 1 ligand, shown here by this little squiggly thing, and we fused this to an FC protein, and then we put an anti-FC IgG along the bottom of just tissue culture plastic. And we showed that if you put cardiac muscle cells, so here myosin is stained in red, and then bromodeoxyuridine for DNA synthesis is shown in brown, you can get proliferating cardiomyocytes. And if you measure the rates of proliferation that are induced by various doses of ligand on the surface, you can see you can get a kind of a bell-shaped dose-response curve where you have a sweet spot where you get close to a doubling of the rate of cardiomyocyte proliferation. So that was cool, but the but is that this was 2-dimensional, life is 3-dimensional, and the second thing is that this only lasts for a few days. It's a relatively short burst of proliferative activity, and then presumably the protein on the surface is degraded. So how do we make this compatible in 3D life, and particularly in the context of stem cell transplantation? So what Caitlin decided to do was to take this anti-IgG that we had and chemically cross-link it to collagen, and then present this in a collagen hydrogel, because we knew that cells like to be in collagen hydrogels. And then she loaded onto this the delta ligand. And you can't simply use delta ligand as a soluble thing, because you need that tension that I mentioned in order to start the whole signal transduction events. And so the notion was that perhaps this hydrogel, this collagen, would hold on tightly enough that it would give the tension that would then expose the cleavage sites, blah, blah, blah. So that was the idea, and this is some work that she did in cell culture where she put an osteosarcoma line that had been engineered to be a notch reporter activity. So the vertical axis is luciferase activity that's an indicator of notch transcriptional induction. And the horizontal axis is simply time. And so you can see when we put it on two dimensions, you get a burst in notch signaling activity, but this peters out and is basically undetectable by 6 days afterwards. Conversely, when we put these osteosarcoma cells into the 3D hydrogel, this signaling period extends out to the better part of, even at 2 weeks, you could still see a little bit of signaling going on. I think the area under the curve was about 4-fold or so increased. So it was a greater burst of signaling and more protracted temporally. So The next thing was, would this work with human cardiomyocytes derived from pluripotent stem cells? And in fact, it did, and we thought it would because it had worked well in 2 dimensions, and Caitlin was a good experimentalist. And so if you compare the IgG-loaded gel, which is essentially empty, and then the one that's been loaded with delta 1, you can see either with young cardiomyocytes, day 15 after induction, we get a boost in their proliferation. And with the older cells, even though proliferation rates are slowing, we still get a boost in proliferation. So this is in 3 dimensions, and that's cool. And then Caitlin went on to show that this gel is injectable into the hearts of athymic rats. And so these are immunocompromised animals that are good hosts for our human cardiomyocytes. This is a picrocereus red stain. So the collagen is red here. The rat myocardium is in green. And this was nice, looked biocompatible, didn't elicit an inflammatory reaction, anything like that. So far, so good. So wanted to, in this, I'm going very fast. This is 3.5 years worth of PhD work, something like this at this point. So we're ready to do the primetime experiment. We always say in vivo veritas, right? And yeah, sometimes, on good days. But this is what we found. We used as our endpoint the size of the human myocardial graft because the whole game was about increasing graft size. So this is the control gel on the left. This is the delta-loaded gel that signals through notch on the right. Each point is one animal. And if anything, the trend went in the wrong direction. So, crap. That was basically our response. We had this, I will never forget this conversation that we had in the office about that. And so it looked like the hypothesis was completely wrong. But the only smart thing that I did in this whole trajectory was to remind Caitlin that she was a very good scientist and that she should believe her data. I said, everything that you did was really solid. We had control of this stuff in vitro. Something's gone wonky in vivo. There is either the system is so flooded with notch that we can't detect a bump in the signal, or there's an antagonist present in our system that we're not looking for. So Caitlin goes and searches the literature and comes back, I think, the next day with this paper that was published out of one of the PLOS journals, and it said cyclosporin disrupts notch signaling. I hadn't had time to explain this to you, but we have a pro-survival cocktail to help keep our cells alive, and we routinely give the animals cyclosporin for at least a week after transplantation. How exactly notch connects to cyclosporin is not clear, but the data look pretty robust. So we went back and did some additional experiments omitting cyclosporin. And this is what we found. Here's histology of, this is an infarcted rat heart that has human cardiomyocytes stained with a human-specific myosin antibody. And this is control graft size. And you can see we had larger grafts histologically. And when we quantify this, we got about a 3-fold increase in graft size after omitting cyclosporin from the cocktail. And if we look at BRD incorporation rates for indices of DNA synthesis, this is a double label between the human-specific myosin and bromodeoxyuridine for DNA synthesis, and these are proliferating cardiomyocytes. And we get more than a doubling in the rate of human cardiomyocyte DNA synthesis. So it looked like we were back on track. So we went on to do a functional study, and this is skipping through about the last 6 or 8 months of Caitlin's PhD thesis to show you the bottom line. On the vertical axis, so this is about 60 rats that went into this particular study. The vertical axis is systolic function of the heart through an echocardiographic index called fractional shortening. It's basically a linear measure of how close the walls come together as they beat. And what we did was to give what we think is a subtherapeutic dose of the cells. So the 2 critical groups to show are these 2. This is the low dose of cardiomyocytes without the delta-1 ligand, and this is the group of cardiomyocytes that had the delta-1 ligand. And what you can see is from baseline, which is post-infarction, but pre-treatment, out to 3 months, there's progression of heart failure in these animals. Same number of cells delivered here, but in the presence of our delta-1 gel, we certainly had no deterioration, maybe a wee bump in cardiac contractile function. We've never seen anything like this with a really puny dose of cells like this. So it looks to be like a fairly robust system to augment cardiac repair. So the conclusions from this first part is that one can engineer a relatively simple but smart hydrogel that promotes notch signaling in 3 dimensions. And one can do this both in cell culture and tissue engineering and in cell transplantation. Used judiciously, this can promote cardiomyocyte proliferation both in vitro and in vivo, as long as one does not have cyclosporin in the system. This enhances graft size, and I haven't had time to show you this, but it also enhances vascularization of the host. So the Notch system is talking to the host, both to the innate immune system and to the vascular cells. And of course, it enhances cardiac function, as I showed you. So we think this might be a nice tool to augment engraftment. And since Notch is widely, it's a powerful system, this may be useful in neurobiology for dopaminergic neurons or for transplanting islet cells and that sort of thing as well. So that's part one. In the last few minutes, what I'd like to do is to tell you a quick story about using cardiomyocytes to repair the heart of a non-human primate and what its functional impacts are. This is work that was done by two talented cardiology fellows, Yen-Wen Liu from Taiwan and Billy Chen from the University of Washington. And this is done in macaque monkeys. We went in with a balloon, with an angioplasty balloon, not to open the artery, but actually to close the artery to create the infarct. And these were large infarcts. We looked at cardiac function by magnetic resonance imaging, and we gave a bunch of cardiomyocytes. And the rest, I think, will be clear as I tell you the story. What I don't know how to do is activate this movie. Can you help me with that, perhaps? There it is. Oh, it's, by golly, did you do that? Oh, some entity did. All right, some sentient creature did. Could you? Let me see if I can click on this. It's hard to find on the. Whoa, it goes fast. There we go. Come on. All right, it's not playing. What you would see if this were playing was a very nice, image of the heart squeezing, and it would just give you a sort of a qualitative sense for how we assess structure and function. The most important slide probably is this one, which we found a robust improvement of monkeys' heart function after we did this. This is a volumetric measure of cardiac function. It's called ejection fraction for the left ventricle, LVEF, and it measures the amount of blood ejected from the heart with each beat. And normal in macaques would be above the scale here. It's about 65%. Baseline is post-infarction, and you can see all the animals had markedly reduced cardiac function, reduced by at least 20 ejection fraction points, and quite a bit of overlap between the treated animals in red and the untreated animals in blue. So the blue is control. And you can see 4 weeks after receiving the human cardiomyocyte grafts, there's minimal improvement in the controls, and there's marked improvement in every one of the 5 animals that received the human cardiomyocytes. Another way to look at this is each animal's individual change in ejection fraction plotted on the vertical axis here, and about a 2.5% in control animals and about an 11.5% in the treated animals. So this is about a 9% delta. I could say, is this a big deal? Is this a small deal? 9% is bigger than anything that has been developed for treatment of myocardial infarction clinically. It's better than reperfusion therapy. It's better than beta-adrenergic blockade, and it's better than angiotensin converting enzyme inhibition. So we think if we could do something close to this in patients, this would be a big deal. The other interesting thing is, well, 4 weeks isn't all that long, and these cells, if you look under the microscope, aren't all that mature. Is there a possibility we'd get increased, that we stop this early? These are really difficult experiments to do because these are chemically immunosuppressed animals. Each one is really individually monitored and so forth. It's like a mini intensive care unit. But we've done 3 experiments where we've taken animals out to 3 months after treatment. And again, the human cardio-treated animals are shown in red, and the control animal is shown in blue. And what you can see is that, again, same thing that you saw at baseline. The animals are comparable, no real change at 4 weeks in the control animal, and about an 8 or a 9 point bump in the animals that received our human cardiomyocytes. But what we saw after taking them out a couple of months longer and letting these grafts mature is we got really very robust improvement in 2 out of 2 animals, both up over 60% ejection fraction. And again, 65 is what we consider the normal baseline for this species. So I've been at this game since 1984 or so, and I have never seen anything in that amount of time that had this impact on cardiac function after a myocardial infarction. I know no reason that this should be better for monkeys than it would be for people. But, there's always a but. The but is that these animals go through a period of electrical instability after we transplant the cells in. Arrhythmias are an intrinsically noisy business. These are the arrhythmias in the control group, shown here as hours per day. And you can see after infarction, before sham injection, and then after sham injection, there's a fair bit of arrhythmic activity in these animals. And here's what happens in the treated animals. And again, if we do statistics, there's no difference. But I want to call your attention to one particular animal, and that's the one shown here. If you follow it back, it didn't have much in the way of arrhythmias prior to transplantation. After we put the cells in, it had this period where it was having almost continuous ventricular tachycardia. This is the Achilles heel right now of the system, and it's the thing that we have to solve before we can take this to the clinic. In an animal with a smallish heart like a macaque, their baseline heart rate's 120, and so bumping up to 180 or 200 is not that big a deal. But in a heart patient like my mother was, somebody getting her heart running up to 180 or 200, I think, would probably be a big deal. So we've got to solve this. So we think that, and we've seen this in other contexts as well to make us know that this is a real thing. So this is, we've got an arrhythmia problem. So we've gone with pediatric cardiologists who are used to doing electrical mapping in small hearts, small primate hearts called children. And they've been able to map the function, the electrical function of the heart. And this is a kind of a weird looking heart because this is low resolution map with this electrode system. But the apex is down here at the lower right, and the base is up here at the lower left. And normally, electrical activation, which is shown, this is a time map for electrical activation, so the first place to activate is in red, should be down here at the apex. And this is one of the animals that came in ventricular tachycardia, and where we're seeing is it's emerging right where we have transplanted some of our human cardiomyocytes, and it's acting like a little pinger, like a signal emitter. So you can see how it propagates out then centrifugally from this one particular focus. And we've done some interventional studies where, I won't go through all the details, but we put it through the paces quite literally. And we expected, let me take a step back. There's two ways that one can get arrhythmias in the heart. One is that you have abnormal conduction of the impulse, and you get a thing that's called reentry. And that's kind of like when people are doing the wave at the stadium. It just keeps going around and exciting itself, that sort of thing. The other approach is that you can have little pingers, a little impulse generation, almost like a pacemaking focus. My money was totally on the reentry problem, but all of the interventions that you've seen here by clinical criterion point against the reentry problem and say that we've got a problem of impulse generation, not a problem of reentry. So it seems like our cells are behaving like little ectopic pacemakers, and for a while there's a struggle over who gets to call the shots on how fast the heart goes. The curious thing is that in culture, these cells are very chill. They never like to go more than 60 or 70 beats a minute. And that's why I thought we were good to go. But something happens when we transplant them and they go crazy for a little while. And that is what we are trying to figure out at the moment. And I'll close with just a couple of shots of histology, since I'm a pathologist and I like histology. This is a low magnification whole slide scan of a control infarct. So collagen is shown, the scar tissue is shown in blue, and then the healthy heart muscle is shown in red with a troponin stain. And there's no human heart muscle to see in this particular one. So what you can see, this is healthy heart muscle over here, and then the wall thins markedly, and we have very extensive infarction and loss of host cardiac muscle. This is what we're seeing pretty consistently in the animals that have human cardiomyocyte grafts. That we've got grafts that's remuscularizing this myocardial infarction, and we have them in the peri-infarct region as well. And we've seen this in virtually every animal that we have transplanted in, ranging from 1 to 3 months post-engraftment. So big, chunky, robust grafts. These grafts are big enough that we can see them by magnetic resonance imaging, so at a macro scale. So to conclude, I'd like to say that you can take stem cells, turn them into a cell type of interest, and here I've told you about cardiomyocytes as a test system, and you can create extensive amounts of new human tissue in an experimental animal model. And I would submit a non-human primate is about as relevant as we could get to a person. There were two things that we were worried about. One of them is teratomas. Would we somehow grow a tumor in the heart of a patient or whatever the target organ is? And it looks like with judicious control of differentiation, this teratoma teratoma thing is under control. I think teratomas probably mean that you didn't do a good job with your differentiation. We remain vigilant. I showed you statistically that we'd had a pretty good improvement of global cardiac function at 4 weeks, and we at least have the potential for further improvement out to 3 months, and that's been in 2 out of 2 animals. We haven't had time to show you, but the subtype of muscle cells that we're making are principally ventricular myocytes. There are different flavors, atrial, ventricular, pacemaker, that sort of thing. The cells that we're making are principally ventricular, and that's what they turn into as they mature. And the biggest rub that we have found so far is this transient pacemaking activity that causes arrhythmias in some of the animals. And so we think that human cardiac myocytes have the potential to improve human cardiac function. And if we're very interested in developing this as a therapeutic and potentially within about a 2-year period doing a first-in-human clinical trial, presuming we can get a handle on the arrhythmic problem. So that's it. Here's the gang that does all the work that I get to stand up and take credit for. I won't give people individual shout-outs because I tried to do that along the way. It's a great group, and I'm very proud to get up and represent their work. And that's it. So thanks very much for your attention.
That's great, Chuck. Does the differentiation in the later times, does the differentiation state go up, or are you just getting more cells? So is the response due to more cells, or is it due to the fact they're finally differentiating much farther along?
We're working on the exact counts, but it's very clear that their differentiation state changes. At 1 month after transplantation, these are scruffy, immature cells with disheveled contractile apparatus, that sort of thing. Their myofibrils are disarrayed and fairly puny. And then they get to be big, chunky, log-shaped cells by 3 months after transplantation, which is also coinciding with the electrical instability versus electrical stability. And so my thought is really that what we're working with is a maturation issue. And if we can get them further down the road before we transplant, it may help function sooner, and it may help rhythm stability.
Hi, Chuck. Peter Zanstra.
Oh, hey, Peter. Nice to welcome back to the Pacific Northwest.
Thank you. I wonder if heterogeneity of the transplanted population might explain the pacemaker-like thing. Do you think just having a more purified or better purified population would help?
I don't know, but I can tell you that when we do our stains, that they turn out to be, at 3 months, they're 99% ventricular myocytes if you use myosin light chain 2V as your arbiter for that. And so I think this, I didn't have time to go into it, but this is with small molecule Wnt modulation for differentiation, and this is all in suspension, bioreactors, that sort of thing. So I think that this protocol, luckily, gives us almost entirely ventricular myocytes to begin with. So that's the best data that I have. I think it's more the heterogeneity of maturity between the graft and the host that leads to electrical instability. And maybe we can chat about some crazy ideas over coffee or something.
So Chuck, to sort of tie the 2 parts of your talk together, you said at one point that the macaques were immunosuppressed.
That's right.
That was cyclosporin.
It is.
So that's one part. And the second part, if you go in and look at the cells that you've transplanted, do they take on more of a sinoatrial node-like morphology, or are they then susceptible? You could use potassium channel inhibitors to try and control the repolarization.
Yeah, good. Both good questions and points, John. So to the cyclosporin issue, I think if we're going to use the notch system, we sure as heck don't want to use cyclosporin. I don't know if that's a general property of, say, calcineurin inhibitors, or is it something? Cyclosporin is quirky in that it has mitochondrial effects and other kinds of things as well. So we need to understand that better. We're also hoping to engineer our cells in such a way that they can be more immunologically stealthy and fly a bit under the immunological radar. People may know David Russell's work that he's doing here. He started a company called Universal Cells to try to make sort of like O-negative blood equivalents for tissue cells that could be used for transplantation. So we're hoping to take some lessons from that. And then the other was the sinoatrial type cells. Are these bona fide pacemakers or not, I guess is another way to say it. I don't think they are. The rub is that immature cardiomyocytes, their natural biology is to have automaticity. And so all cells, atrioventricular and true pacemaking cells, have pacemaking activities in the early stage of development. And then this attribute is lost as the cells mature. And so the ventricular cells in you or me would be quiescent at this point. But that's a developmentally later phenotype that results in rearranging your ion channel gene expression and that sort of thing. And we are working with every pharmacological agent under the sun right now, but it's too early to say what the answer is. Potassium channel blockers are a good idea, though.
So Chuck, really great work.
Hey, Sean.
So you're transplanting the cells into a pretty hostile environment with scar tissue and a lot of other problems. You seem to be getting fairly good survival, though. Do you think it'll be useful to also put in stromal cells, endothelial cells to help that?
That's a great question, Sean. We have another paper that's currently in rejection that is looking at just that. And what we took a page out of your book where we made epicardial cells, and we have co-transplanted them with cardiomyocytes because the epicardium developmentally is the progenitor for all the stroma of the heart, the fibroblastic type cells and that sort of thing. And we see threefold enhancement in graft size when we use these epicardial cells. So maybe.
Well, at least you're getting to the paper stage. My grants are under rejection.
I think you've got one more.
Hey, Chuck. I was wondering in the first part when you did the no cells control with the collagen gel with Delta, it looked like that also had an effect. And
so that's a great. So there, where's Caitlin? She's scurried away. So we're talking about that. Is there some biological effect to that? Maybe is the answer, and we underpowered those two gel-only groups a little bit just because we were trying to get the dang project done. And we thought that they should be so uniform that we should have small variance. I think in retrospect that was a mistake. So we're going to go back and do some additional ends. So you were watching carefully. That's so good on you. And that's also something that our reviewers pointed out to us as well. And so maybe, and actually you could say, oh, that's a bummer for your story, maybe. But it's also it's a cool thing if signaling to the host through Delta has salutary effects. I mean, terrific, right? And so we can just tell a little bit different story if indeed that's the way it pans out. Thanks. All done?
Great.
Good.
Let's
Thank
thank
you.
our morning speakers.